Now let's return to the target ID. We've already looked at a kinase, HCK, so now we wanted to investigate some enzymes as well, and they're important biological catalysts that are often targeted in cancer treatment. On the screen, you can see the new ranking of genes. As the Pandomics target ID was updated throughout the last few weeks, our top genes were different, and in our old list, we analyzed the first 10 to 15 genes by looking at literature. Among these, many genes were downregulated, so we discarded them, but out of the upregulated ones, there were some really interesting ones. For example, TP53I3, also shown up here, it's part of the oxidoreductase family, and there is evidence that suggests this gene is a prognostic biomarker for glioblastoma, but that's basically all about its relation to glioblastoma. Moreover, there are no small molecules developed for this gene. ALKBH2, the sixth gene, also seems interesting at first. It is too part of the oxidoreductase family and it's upregulated. However, we still don't really understand its true functioning in relation to glioblastoma, and there has only been one publication about it. It was in 2013, which is quite a while ago. The other top genes were mostly downregulated in glioblastoma cases, so we didn't look at them. And out of here, we chose MCM8 and EME1. MCM8 is related to the pathway activation of ATR in response to replication stress, and I looked at this pathway in Pandomics and found that it was significantly activated in glioblastoma samples in several different datasets. This provided additional evidence for MCM8 being an important biomarker in glioblastoma. MCM8 has a high Pandomics score for network neighbors, pathways, interactome community, and expression scores, and it was ranked as number 2, as I mentioned. Our dataset box plots show that the expression of this gene is increased in 9 out of 10 datasets, and this opens up the possibility of MCM8 being a biomarker for glioblastoma. MCM8 is part of the MCM family, and it's a component of the MCM8-MCM9 complex, which repairs DBSs and DNA ICLs by homologous recombination. It is also important to understand for DNA resection of the mRNA complex, as it recruits it to the repair site and promotes the complex nuclease activity. This gene is localized in the nucleoplasm and has a low RNA tissue specificity. The most important idea to take out of this is that MCM8 is part of a repair system, which is something cancer cells often acquire and ameliorate. We hypothesize that since this gene is upregulated in glioblastoma, based on literature you will see in just a minute, there is a chance of slowing down glioblastoma development by downregulating this gene. However, since this gene is involved in the cell cycle, we should investigate the safety of inhibiting it first, as studies have shown that mice with inhibited MCM8 are sterile because spermatozoids are blocked in meiotic prophase 1, and females have only arrested primary follicles and frequently develop ovarian tumors. And MCM8 lacking embryonic fibroblasts show growth deficits and chromosomal damage and cannot overcome the transient inhibition of replication fold progression. So on the images on the right, you can see RNA data from the Human Protein Atlas. What's important from these figures is the high expression of MCM8 in the brain. MCM8 is involved in the progression of cell cycle pathways, as shown in the image. The whole MCM family contributes to promoting the cell cycle and knocking it down. MCM2, MCM3, and MCM6 specifically caused cell cycle arrest or delayed progression at certain points. An example of MCM8's role is that when it's bound to cyclin D1 to activate RB protein phosphorylation by CDK4, the binding allowed the cell to enter into S phase. A study found that a heightened expression of MCM8 increased cancer recurrence in patients with different cancers, and that forcing more expression stimulated fast cell growth and transformation in xenografts. On the other hand, knocking this gene down caused cell growth arrest and decreased tumor volumes and mortality of severe combined immunodeficiency mice, xenografted with glioblastoma cells. It was also shown that patients with prostate cancer recurrence, which was the main cancer studied in this investigation, also gained MCM8 copies. The conclusion was that the copy number increase and overexpression of MCM8 may play critical roles in human cancer development. On this slide, figure C is a Kaplan-Meier graph showing the difference in survival for those with MCM8 expression of over 2 and under 2. Figure D shows the survival rate difference between patients with MCM8 copy gain and with no gain. There are currently no drugs for MCM8, however, the attention to this gene has been rising over the past few years, and you can see this by the trend in attention scores going up in Pandomics. On the other hand, Pandomics predicts that there is quite a low chance that a drug targeting this gene will go through to phase 1. Let's look at the final gene, EME1. It is on the same filtered score as MCM8. As you can see, EME1 ranks quite highly here, and it's on the 7th place, whereas without filters, it used to be on the 95th spot. EME1 is part of the HDR through homologous recombination pathway. We can see that it is highly upregulated, which suggests furthermore that EME1 is a good potential target. We also made box plots for EME1, and as you can see, in 9 out of 10 of our datasets, there is a higher expression of this gene in glioblastoma patients as compared to control patients. EME1 belongs to the EME1 MMS4 family, and it interacts with MUS81 to form a DNA structure-specific nuclease with substrate preference for branched DNA structure with the 5' end at the branch nick. It is localized in nuclear bodies and most likely in cytosol as well. You will notice from the images on the slide that it has a high protein expression almost everywhere, while the RNA analysis show that the highest expression is in blood plasma. As for protein expression, what's the most important for us is that it is expressed in the brain, as we're specifically investigating glioblastoma. And due to the blood-brain barrier, we can inhibit this gene specifically in the brain. EME1's role in cell signaling is shown in this photograph. It models how when the cell cycle arrest occurs for any given reason, an EME1 protein binds to the part of the DNA structure which caused it. And this brings endonucleases that cut open the DNA segment and promote further binding of other molecules that aim to repair whatever caused the arrest, allowing the cell to move further along the cell cycle. Thus, inhibiting EME1 would stop the cell from moving on in the cell cycle, which could potentially prevent cancerous proliferation in glioblastoma cells. A study was conducted with Jordan patients on EME1 expression in glioblastoma, and to be more specific, they wanted to find an association between some genes, including EME1, EXO1, ERCC2, and BRCA1. They extracted DNA from all their patients. Afterwards, they set up a correlation between the survival rate of the patients and their genetic codes to see whether a specific sequence could augment the risk of acquiring glioblastoma. The graph made by the study shows how having specifically 2 cytosines in this gene that encodes EME1 seemed to be more beneficial than having 2 thymines or a thymine and a cytosine next to each other. They also came to the conclusion that in the nucleoside excision repair pathway, EME1 endonuclease is essential to overcome any blocks in DNA replication through processing the stalled replication forks. From this, we can conclude that inhibiting EME1 in cancer cells will slow down nuclear excision repair and lower the survival of these cells. Currently, there are no drugs against this gene, and the attention score is mediocre, and the trend to look at this gene has actually gone down. This, on the other hand, might not seem that good at first, but since this gene is not that well known, yet it plays such a great role in glioblastoma, it might be a good therapeutic target to prevent DNA repair and cell division in cancer. It's an endonuclease from the hydrolase family belonging to esterases, and these are normally well-targetable genes.